A very good morning to all of you. Today we are going to discuss the physiology of blood. At the end of this lecture, you will be able to list the components of blood. You will be able to differentiate the plasma and serum. You will be also learning about the different types of plasma proteins present in the blood. And also, we will be also explaining you about the hemopoiesis, the steps involved, and the role of the erythropoietin in erythropoiesis. Blood is considered to be a fluid connective tissue which connects one organ to another. You'll be seeing that blood is flowing through various parts. It is connecting different organs. For example, kidney is connected to the heart through the blood vessels, and blood is the mediator in between. Approximately 8% of the total body weight is contributed by the blood. If you look at the average volume, approximately 5 liters is seen in the case of women and 5.5 liters of blood is seen in the case of men. The slight difference, slight higher value in the case of men is seen because of high production of RBCs. We'll be discussing about this changes or what is the difference in the case of males and females when we discuss about the erythropoiesis. 55% of the blood is plasma and 45% is made up of formed elements. Formed elements means it is the cellular component of the blood. For example, RBCs, WBCs, platelets, etc. These cells are evenly dispersed in the plasma and the pH is about 7.35 to 7.45. The pH of the blood has to be maintained constant in order for the enzymatic reactions to happen in an optimum way. The functions of blood includes transportation of various gases like oxygen, carbon dioxide, and also transportation of metabolic waste products from the excretory organs, and it will be excreted outside the body. Transportation of various nutrients to different parts of the body, carrier for various hormones. Even in endocrinology, we have seen that various hormones are transported in blood because it is directly secreted into the bloodstream. It has an important role in the homeostasis for regulation of the extracellular fluid volume, for the regulation of pH, temperature, and osmotic pressure. Blood also contributes for the protective function of the body from foreign microbes. For example, WBC provides immunity, and also we'll be looking at again one by one what are the different functions. Hemostasis is the process by which it will be preventing the blood loss after an injury. That's also called as a coagulation. The whole blood is divided into plasma and formed elements. Plasma is the fluid portion of the blood, whereas the formed element is the solid portion of the blood or the cellular element of the blood. If you carefully observe plasma, it has got dissolved proteins which are called plasma proteins, and the three major plasma proteins what you see, they are albumin, globulin, and fibrinogen, and which will be having different functions which will be explained in the next slide. The plasma also has amino acids. It will be also carrying nutrients. It will be carrying the blood gases. It will be having different types of electrolytes and also the nitrogenous waste which will be excreted out from the excretory organs. It will be passing through the blood and it will be taken through the excretory different organs. And if you look at the formed elements, the major formed element is the erythrocytes or the red blood cell. Then we have platelets. Then we have leukocytes, different types of leukocytes. How to differentiate plasma with serum? Now, if you take a sample of blood and if you put it in a test tube, you can see in the left hand side a test tube in which anticoagulant is poured along with the blood. What is an anticoagulant? Anticoagulants are those substances which will prevent the blood from clotting. That means if the blood is mixed with an anticoagulant, the blood will not clot. So if you allow that blood to stay in the test tube, it will not clot. In this case, what is done is an anticoagulated sample of blood is taken in a test tube. It is centrifuged in a centrifuge machine for 3000 rotations per minute for about half an hour. You get 3 different layers like this. You can see, you can appreciate that there is an upper layer, a yellow colored layer, which is the plasma layer. Then you have a layer in middle, which is called as a buffy coat, which consists of WBCs and platelets. And you have a lowermost layer, which is usually consisting of only red blood cells. So the lower layer is the RBCs. The middle layer is the buffy coat, which consists of platelets and red and white blood cells, and the upper layer, which is having only the plasma. Now, if you keep the blood test tube without adding an anticoagulant, look at this right side. This is a blood kept inside a test tube without adding any anticoagulant. What will happen? It will definitely clot because there is no anticoagulant in it. So in this test tube, you can appreciate that the clot is formed at the lower end. And once the clot is formed, there is a fluid which is oozing out of the clot. This clear fluid, that is called as serum. So the serum is the liquid part of blood after the coagulation. Therefore, it is devoid of clotting factors such as fibrinogen. Whereas in the case of plasma, plasma is also a liquid portion, but it still has the clotting factors in it. In the serum, the clotting factors are absent because these clotting factors are used up during the clotting process. So in the right hand side, the picture also you can see that this is a clotted blood. So you can see this is the blood clot. Everything is clotted, and here the clear fluid above, that's the serum, and that doesn't contain the clotting factors. Now we will see some of the important definitions. Hematopoiesis or hemopoiesis, it is the formation of blood cells which will be occurring in the bone marrow. Hematocrit or packed cell volume. When you see in this particular test tube in the right hand side, the lower portion is composed of only red blood cells or erythrocytes, which is called as the hematocrit, or it is also called as a packed cell volume or PCV. If you see the PCV values, in the case of males and in the case of females, it is totally different. You can see that in the case of males, adult male, it is 45 plus or minus 5%, whereas in the case of an adult female, it is 42 plus and minus 5%. Now, can you think of why in the case of females, the hematocrit or packed cell volume is lower? The answer is simple. They are having low RBC count when compared to the males. The males are having high red blood cell count when compared to the females because of the hormone testosterone. The male hormone testosterone has a stimulatory effect on the red bone marrow to produce more red blood cells. Now we'll see the bone marrow. Can you appreciate these two pictures? The left hand side, you can see the skeleton of a newborn or an infant, and you can see in the right hand side the skeleton of an adult who is 25 years or more than that. What is the major striking difference what you see in these two skeletal systems? You can see that the red bone marrow is located. What is red bone marrow? Red bone marrow is the one which is forming the red blood cells. You can see that in the case of an infant skeleton who who is less than 1 year, all the bones in his body is made up of red bone marrow, which means each of these red blood cell can be produced. Each of these bone marrow, red bone marrow, can produce the red blood cell. That is why you will be seeing a very high red blood cell in the case of an infant. Whereas in the case of an adult, most of these red bone marrow will get replaced into yellow bone marrow due to accumulation of fat. You can see in this picture, the red bone marrow is restricted to only to the tips, whereas the central areas, you can see that it's mainly of the yellow bone marrow. So these are the locations where the red bone marrow is restricted in adult, which is the cranium, sternum, ribs, pelvis, and the upper end of long bones. So if you want to get a sample, how can you get in an adult? So you need to take a sample by using a procedure which is called as the bone marrow biopsy, using an aspiration needle, and usually the iliac crust. You can see that this is the iliac bone, and the iliac crust is the preferred location through which you can draw using a needle. You can take a sample of bone marrow, because in this picture also you can see that in the iliac bone it is having the red bone marrow. So in the case of an adult individual, you can see that the red bone marrow is restricted to only these parts, whereas the other bones, the red bone marrow will be replaced by yellow bone marrow. Now I would like to highlight one more important point. Why there is a difference between the red blood cell production in the case of an infant and an adult? Which one do you think that the red blood cell production is higher? Of course, in the case of an infant, the RBC productions will be greater, because you can see that all bones are made up of red bone marrow. Whereas in the case of an adult individual, you will be seeing that it is restricted to only specific location. That is the reason when you compare an infant with an adult, always the infant's red blood cells count will be higher when compared to the adult. Now coming on to the components of the blood, you will be seeing that the plasma is the major contributor, that is 55% of the blood, which is the fluid portion of the blood, and approximately 45% of the blood is made up of formed element, which are the cellular portion. So in the case of plasma, you can see that water is contributing to about 90%. See here, 90% is contributed by water. Solutes will be 10%, and here all the respiratory gases, etc., will be dissolved in these fluid portions. And also you will be seeing that it will be carrying hormones, electrolytes, and also it will be consisting of plasma proteins like albumin, globulin, fibrinogen, etc. In the formed elements, 45% is contributed by the formed elements, which are the cellular portion of the blood. Here you can see it is erythrocytes, which are the red blood cells, which is the major contributor. Then you can see different types of leukocytes, which are the white blood cells. Leuko, which means white. Erythro, which means red. And you can also see the platelets. So these three types of cells which will be contributing to the formed elements. Coming on to the plasma proteins, 7 to 8% of plasma volume is contributed by the plasma proteins. And one of the important factor which is contributed by the plasma protein is the osmotic pressure. It is the presence of the osmotic substances in the plasma, right? In this, albumin is the major contributor. Albumin is one of the major plasma protein which is contributing to the osmotic pressure, which maintains the water balance between blood and tissues. This is the force that determines whether the blood has to lie inside the blood vessel or it has to go to the interstitial space. It is involved in pH buffering and clotting of the blood, and also various defense mechanisms like in the forms of antibodies. It is also involved in lipid transport. The three major types of plasma proteins: albumin, which is abundant, which is also having a role in the pH buffering system, which is involved in the colloidal pressure, which is involved in the colloidal osmotic pressure, which is helpful in transportation of insoluble substances. Then you can see globulin, which has got alpha and beta globulin, which will be transporting insoluble substances and also the hormone precursors. One of the example is angiotensinogen. Then you can see the gamma globulins, which are providing the immunity, which are made by the lymphocytes. Fibrinogen is another plasma protein which will be contributing to the blood clotting process. All plasma proteins are synthesized by liver except gamma globulins. That's the reason when you have a liver disease, when a patient is suffering from liver disease, the production of all plasma proteins are affected except the gamma globulins. You can see that all the plasma protein production will be affected in the body because the liver is the source of these plasma proteins. Now we shall discuss upon the red blood cell or the erythrocytes. It is one of the major component of the hematocrit or the packed cell volume. You can see that they are small in size, they are anucleate. That means a mature red blood cell doesn't contain a nucleus. When you look at the shape, it is having a characteristic shape. It is a biconcave shape. That means the center portion is depressed a bit, and from the sides, if you see, it appears exactly like a dumbbell, right? It is a kind of a dumbbell shaped. That means it is having a central area which is a biconcave disc-like shape. They contain hemoglobin, which is the iron-bearing protein that will be helping the RBCs to transport oxygen to various parts of the body. Because of their unique shape, these RBCs are ideally suited for gas exchange. You can see that, as I mentioned, this is having a dumbbell shape. This shape also helps these RBCs to literally squeeze through the small pulmonary capillaries. If you see the pulmonary capillaries which is there in the lungs, and when the blood flows through the lungs for oxygenation, actually the size of the diameter size of these capillaries are much smaller compared to the size of the red blood cells. But in order to accommodate or get into or inside these small pulmonary capillaries, literally these red blood cells have to squeeze through the capillaries. So this particular type of shape or this characteristic shape help these RBCs to get adapted and to squeeze through small diameter pulmonary capillaries. RBCs doesn't have a mitochondria and generate ATP anaerobically. They will be producing ATP anaerobically, so they do not consume any of the oxygen which they are transporting. The main function, if you look at it, they are transporting the oxygen, but they do not consume the same oxygen which they are transporting because they do not have mitochondria. And the normal count, you can see that this is the normal count, it is millions. Excessive number of red blood cells, which is called as a polycythemia, more than the normal range. If the red blood cells are increasing in production, and if you see a high number of red blood cell in the blood, that condition is called as a polycythemia. And if you see that the number of cells is decreased, that condition is called as anemia. Please remember that anemia doesn't mean that it is the only reduction of the red blood cells. It can be also due to a reduction in the hemoglobin content, right? So anemia means it can be a reduction in the RBCs, or it can be a reduction in the hemoglobin content in the RBCs. For example, 5 million cells are present. The RBC, the number wise, it is fulfilling the criteria. But imagine if there is less number of hemoglobin in each RBCs. So again, the person or the patient is called as an anemic because he is having less oxygen carrying capacity. So anemia means it can be a reduction in the red blood cell count itself, or it can be a reduction in the hemoglobin content. The normal hemoglobin concentration in male and female and newborns. You can see that in the case of males, the hemoglobin is about 13 to 18 gram per deciliter. Deciliter means it is per 100 ml. In the females, you can see that it is 12 to 16 gram per deciliter. 12 to 16 gram per 100 ml. And in the case of a newborn, it is 18 to 20 gram per 100 ml. Now, I want you to think on these three values. Why there is a difference in all these three individuals, males, females, and newborns. Why in the case of newborns it is very high? We have seen when I discussed about the red bone marrow, I told you that in the case of a newborn, all the bones in the body is having red bone marrow. That is the reason why you are seeing higher RBCs, and when the RBCs are higher in number, you can see that this particular RBCs, or if it is higher, you can see that the hemoglobin content or the concentration also will be higher. In the case of females and males, when you compare, if you take a comparison, again, in the case of males, you can see that the red blood cells are higher. That is why you are seeing the hemoglobin concentration in the case of male is also higher. It is 13 to 18 when compared to female, which is 12 to 16. And hemoglobin consists of 2 alpha chains and 2 beta chains. I am not going into the details of the hemoglobin as you have already learned it in the biochemistry. And it is the center of each heme group, the atom of iron, which binds to one molecule of oxygen. These are the normal levels of hemoglobin in males, females, and in newborns, and these values, you need to memorize these values because it is important. We will be now looking at what are the different forms of hemoglobin. Oxyhemoglobin, which is appearing ruby red in color. Whenever hemoglobin is in combination with oxygen, that is called as oxyhemoglobin. That's the major form of hemoglobin which is seen. 98.5% of oxygen in blood is carried in the bound form along with hemoglobin. Now we will see the second part, that is the deoxyhemoglobin. Whenever the oxygen detaches from the hemoglobin, called as a deoxyhemoglobin or reduced hemoglobin, which appears dark red in color and which will be seen in the veins. The third type of hemoglobin what is seen is called carbaminohemoglobin. Whenever the hemoglobin binds with carbon dioxide, this occurs when hemoglobin is in the reduced state and when it picks up carbon dioxide from tissues and delivers it to the lungs. Hemopoiesis or hematopoiesis is the process by which the blood cells are formed. All formed elements arise from pluripotent hematopoietic stem cell, the hemocytoblast. Hemocytoblast is the pluripotent hematopoietic stem cell, which when received by an appropriate stimulus, it can get transformed into red blood cells. It can transform into various types of WBCs and so on. The number of erythrocytes in the blood is constant due to a balance between RBC production and RBC destruction. Erythropoiesis is the formation of new red blood cells. The process begins with the stem cells or the hemocytoblast. It takes approximately 7 days to form reticulocyte, which is an immediate precursor cell of a mature RBC, which means it will take about 7 days to form a cell called reticulocyte, which is an immature RBC, which is just a precursor of the RBC, precursor of a mature RBC. Just before it forms the mature RBC, that stage is called as a reticulocyte. So it will be taking approximately 7 days for forming the reticulocytes. And reticulocytes will leave the bone marrow and enter into the blood, and in the blood they get matured. So it will be taking approximately another 2 to 3 days to mature into a mature RBC. Takes about 2 to 3 days to mature into erythrocyte. That means the total erythropoiesis, you can see that in this case 7 days for the formation of reticulocytes and 2 to 3 days to form from the reticulocytes to get transformed into erythrocytes. So total it is taking approximately 10 days, 7 plus 3, 10 days you will take approximately to get the mature RBC. Remember, the mature RBC is anucleated, it doesn't have a nucleus. Now we will see the steps involved in erythropoiesis. Erythropoiesis is the formation of red blood cells from a hemocytoblast, which is a stem cell. Right from the stem cell, this is the stem cell here, the starting cell, and when you see across all these stages, what is the most striking feature what you see? Of course, it is the size of the cell. The size of the cell is very big when you are looking at the stem cell, and when you are looking at the erythrocyte, the size becomes smaller, right? The size reduces. You can always see a definite nucleus when it was immature as a stem cell. You can see nucleus is seen all over, nucleus is seen, and finally at the stage which is called normoblast, where you will be seeing the nucleus is being extruded out. Extrusion of nucleus will take place at the stage of normoblast. That means a cell was there with a proper nucleus, and as it matures, the nucleus is extruded out of the cell. So at the stage of this particular stage, which is a normoblast, you will be seeing that the cell will be losing the nucleus. Why it is losing the nucleus? Because we want the red blood cells to hold more more hemoglobin. The red blood cells should hold more hemoglobin, and that's the main function. The main function of the red blood cell is to carry oxygen, right? So for that it requires hemoglobin. So for keeping hemoglobin, it requires a big space there. So we don't want that nucleus occupying that place, so that has to be extruded out. Then you can see the reticulocyte. Reticulocyte is the immediate precursor of that of a erythrocyte. Erythrocyte, this one, the last one, is the mature red blood cell, which is small in size, without a proper nucleus, without a nucleus, because nucleus is already extruded out from during the normoblastic stages, right? And also you'll be seeing the staining changes. The color of the cell changes from a blue colored, and slowly it become a reddish. You can see that the hemoglobin will start appearing in these stages, and slowly as the hemoglobin start appearing inside, the cell color changes, and finally it will be totally red in color when it appears as a mature erythrocyte. Red blood cells normally is replaced at the rate of 2 to 3 millions per second, and the average lifespan is about 120 days. After 120 days, each red blood cells will be destroyed in the reticuloendothelial system, especially in organs like spleen. In this slide, you will be seeing the life cycle of the red blood cell. As you know, the lifespan of a mature RBC is 120 days. After 120 days, what happens to the red blood cell? Definitely, it will be going into the spleen, and broken down by the reticuloendothelial system. And what will happen to this components of the hemoglobin? You look at this picture, you can see that hemoglobin will be broken down into the heme group and the globin group. In the globin group, it will be further divided into or broken down into amino acids, and it will be absorbed back into the bloodstream. Whereas the heme portion, you'll be having a bilirubin, which is the pigment which will be picked up by the liver. There will be an iron portion which will be then stored in the form of ferritin or hemosiderin inside the liver, and you will be seeing that the bilirubin will be then secreted into the intestine in the bile. Bile, that green color of the bile is due to the bilirubin, the pigment bilirubin, and then it will be metabolized by the bacteria in the intestine, and it will form the stercobilin. Stercobilin is usually excreted out in the feces, right? So that is how the life cycle will be taking place, and the raw materials are made available in blood for the erythrocyte synthesis. All the raw materials, whatever is reabsorbed, it will be used again into the bloodstream for next making of the next red blood cells. In this slide, you will be looking at what is the exact role of the hormone erythropoietin, which is secreted from the kidney, and what is its role on the erythrocyte production. Once again, recall the location where the erythrocytes are produced. Erythrocytes are produced inside the bone marrow in an adult. So now let us see what is the role of erythropoietin. Now look at in this particular picture. In the kidney, in response to the reduced oxygen carrying capacity, whenever a situation of hypoxia arises, hypoxia means a low level of oxygen in the blood, and it will be delivering low oxygen to the tissues, right? Any of those situations, it is called as hypoxia, where the tissue is suffering from low amount of oxygen. So the kidney cells will be stimulated, and these cells will be secreting the hormone erythropoietin. Erythropoietin is released from the kidney, and it will be stimulating the bone marrow where the red blood cell production can be increased. Now what will happen when red blood cell production increase? In the red blood cells, there is hemoglobin. Hemoglobin can carry oxygen. So here the stimulus was a reduction in the oxygen level of the blood, that is hypoxia. Hypoxia is the stimulus. As a result, more erythropoietin is released from the kidney. It will go and stimulate the red bone marrow to produce more and more red blood cells, and red blood cells will be carrying oxygen. So the oxygen carrying capacity is increased. So this is an example for a negative feedback mechanism. So here also you can see that the kidney detect reduced oxygen carrying capacity of the blood. When oxygen delivery is low, the cells in the kidney secretes erythropoietin. More erythrocytes are formed and therefore increase in the oxygen carrying capacity, and there is a relief of the original stimulus. This slide also indicates the action of erythropoietin and how it regulates the. So look at the beginning, normal oxygen levels. Now here, whenever the normal oxygen levels are disturbed, like in cases as a stimulus is hypoxia due to decreased RBC count or decreased availability of oxygen in the blood, or an increased tissue demand, all these things will be creating hypoxia. These oxygen levels, low oxygen levels in the blood, will be when it is going through the kidney. When the blood is going through the kidney with a low oxygen carrying capacity, the kidney cells will stimulate the hormone erythropoietin. Erythropoietin will be released from the kidney, and then it will be stimulating the red bone marrow, and finally more RBCs are produced, and increasing the oxygen carrying capacity, and it will be normalizing the oxygen levels. Now let us look at the factors that enhance erythropoiesis. it is classified under nutritional factors, which include iron and amino acids, which are usually supplemented as a part of nutrition of the body. There are maturation factors. The most important maturation factors are vitamin B12, and vitamin B12 is essential, or it is needed to stimulate the formation of thymidine triphosphate, which is the building block of DNA. And for the folic acid. So these are the two maturation factors, which are vitamin B12 and folic acid. In case of vitamin B12 deficiency or folic acid deficiency, there will be a problem with the maturation of the red blood cells. Usually the blood cells which is released into the circulation will be in an immature state if the individual is having vitamin B12 deficiency or folic acid deficiency. The maturation process is not completed because they are important maturation factors essential. And normally the folic acid is obtained from green leafy vegetables, some of the fruits, liver, etc., and they are destroyed during cooking. Controlling factors for erythropoietin is the main thing, the erythropoietin, the hormone which we discussed 2 slides ago. That is the hormone which is released from kidney, the erythropoietin, which is the major hormone which will be contributing to erythropoiesis. And a healthy bone marrow. Yes, a healthy bone marrow is essential, because in case if the bone marrow is exposed to harmful radiations like X-rays, mostly there is a depression in the production of the red blood cells. The erythropoiesis will stop. So those kind of anemias which is usually developing as a result of a depressed bone marrow, as a result of, like for example, X-rays, those kind of bone marrow depression is called as aplastic anemia. The bone marrow will not be able to produce enough amount of RBCs and the individual will have anemia.